We'll start the presentation. Good afternoon to everybody, and thank you for and most of you are coming in. A few more to come in. I think will come a little later. So today's presentation is on hemoglobin. Last time we spoke in terms of the hematology, and I think we covered hemoglobin and the WBC, all the others, and we spoke a little on anemias and stuff like that. So today let us focus on the most important parameter and protein that you and I have, because without that you and I would be in trouble, and that is hemoglobin. So in this presentation we will review hemoglobin and its structure, synthesis and function, different types of hemoglobin and variants which are very important to differentiate, and type anemias, especially the inherited ones, uses of hemoglobin and its relationship with hematocrit and red cell. You will review some results on our magnificent machines. This will be a discussion with our experts a little later. This will help you to help your clients better. You will review HbA1c and its relationship to diabetes control. Finally, you will review heme synthesis, its build-up and breakdown, and how important it is that it happens at the right time. So we take a look at a little three-dimensional picture of hemoglobin. So hemoglobin, we usually call it HGB or HB. It's an iron-containing oxygen transport metalloprotein in the red cell of almost all vertebrates as well as tissues of some invertebrates. Hemoglobin in blood carries oxygen from lungs or gills to rest of the body. There it releases the oxygen to permit aerobic respiration to provide energy to power the functions of the organisms in the process called metabolism. So here you had a picture of the hemoglobin. Let's go back to that. Right? So we've got somebody's coming. Okay, let somebody come in. This is what happens in between over here, and we please do excuse us for something like that. No name. Urvi is coming. Okay, admit her. All right. All right. All right. We're coming back to the second slide. So we took a look at the red cell. We took a look, and we will be taking a look at the structure of hemoglobin and heme. So hemoglobin is involved in the transport of other gases. Guys, this is very simple. You and I know this. I think kids in the second and third and fourth standard are taught this. It carries some of the body's respiratory carbon dioxide, about 20-25%, as carbon hemoglobin, in which CO2 is bound to the heme protein. The molecule also carries the important regulatory molecule nitric acid bound to a globin protein thiol group, releasing it at the same time as oxygen. Excessive glucose in one's blood can attach to hemoglobin and raise the level of HbA1c. Hemoglobinemia is a medical condition in which there is excess of hemoglobin in the blood plasma. This is an effect of intravascular hemolysis, in which hemoglobin separates from red cells and can lead to anemia. You're continuing with hemoglobin. So it's synthesized in a complex series of steps. Those of you whom I've taught, I don't know whether it's clear in your mind, these biochemical steps which are so important and interesting. The heme part is synthesized in a series of steps in the mitochondria and the cytosol of immature red cells, while the globin protein parts are synthesized by ribosomes in the cytosol. Production of hemoglobin continues in the cells throughout its early development, from proerythroblast to the reticulocyte in the bone marrow. At this point, the nucleus is lost in mammalian red blood cells, but not in birds or many other species. So here you have, on the right-hand side, you've got the oxygenated hemoglobin molecule, and we've given a little bit of a substance in the middle, how hemoglobin is carried. Even after loss of nucleus in mammals, residual ribosomal RNA allows further synthesis of hemoglobin until the reticulocyte loses its RNA soon after entering the vasculature. The hemoglobin synthetic RNA, in fact, gives the reticulocyte its reticulated appearance and name. Now, towards July, we have Mr. Jaldeep Bansali, who's going to talk to you in terms of the reticulocyte and how important it is and how it is picked up on the machines today. We're also very happy to have Sandhya with us, who is going to take the last lecture, and she is going to tell you all about the hemoglobin, the red cell, and so on the machine. In general, hemoglobin can be saturated with oxygen molecules, oxyhemoglobin, or desaturated with oxygen molecules, desoxyhemoglobin. Oxyhemoglobin is formed during physiological respiration when oxygen binds to the heme component of the protein hemoglobin in red cells. And of course, I think you all very familiar with this little instrument, especially when we had COVID times and our hemoglobin, our oxygens were checked. So there's a little bit of note up here. Deoxygenated hemoglobin is a form of hemoglobin without the bound oxygen. The absorption spectra of oxyhemoglobin and deoxyhemoglobin differ. The oxyhemoglobin has significantly lower absorption of the 660 nanometer wavelength than deoxyhemoglobin, while at 940 nanometers, its absorption is slightly higher. The difference is used for the measurement of the amount of oxygen in a patient's blood by an instrument called the pulse oximeter. This difference also accounts for the presentation of cyanosis, the bluish or purplish color that tissues develop during hypoxia. And I think you all know the meaning of hypoxia, less hemoglobin. So we come to the types of hemoglobins. Hemoglobin variants are a part of the normal embryonic and fetal development. There may also be pathologic mutants form of hemoglobin in a population caused by variation of genetics. Some well-known hemoglobin variants, such as sickle cell anemia, are responsible for diseases and are considered hemoglobinopathies. Other variants cause no detectable pathology, but are thus considered non-pathological variants. And we are going to talk a lot in terms of sickle cell anemia, thalassemias, and a little bit of G6PD also a little later. So now here we go, there are more than 750 variants of human hemoglobin have been identified, many of which give rise to hematological disorders. And we are going to talk about a few of them. So this is what starts off like in the embryo. You got GABA 1, GABA 2, hemoglobin Portland 2. In the fetus, it's hemoglobin F, alpha 2, gamma 2. After birth, hemoglobin A is alpha 2, beta 2. That's the adult hemoglobin. The most common with a normal amount of over 95% in our blood cells. And you can take a look at the pictures on the right-hand side. You've got normal hemoglobin A, alpha 2, beta 2. You've got fetal hemoglobin alpha 2, gamma 2, and you got A2, which is your delta, alpha 2, delta 2. So alpha 2, delta 2 gamma synthesis begins late in the third trimester, and in adults, it has a normal range of 1.5 to 3.5. So this is your delta, alpha 2, delta 2. So this usually, I think, is associated with a certain type of thalassemia, the heterozygous thalassemia. Hemoglobin F fetal hemoglobin alpha to gamma 2 in adult hemoglobin F is restricted to a limited population of red cells called F cells. However, the level of hemoglobin F can be elevated in persons with sickle cell disease and beta thalassemias. And you can take a look at the table on the right hand side. Use of hemoglobin. Normal values of hemoglobin in the first and third trimesters of a pregnant woman must be at least 11 grams per deciliter or at least 10.5 grams per deciliter in the second trimester. Dehydration or hyperhydration can greatly influence measured hemoglobin levels. Albumin can indicate hydrated status. Somebody coming in? Just a moment, guys. Please excuse us. Somebody's walked in.
Excuse me, the slides are not changing. Can you check, please?
Shanti, that is the reason why we've given you the given you the PPT to keep it in front of you, because
Okay.
this is the problem with Google. That's the reason why we've sent the PPT in advance. Some of you
Okay.
may be able to see it, but on the whole, it does not show. And we've asked people, and they think this is the way it is. So please follow the PPT. Who's coming? All right, shall we go further? You'll have to excuse us for that. On what page are you at? Shanti, what page are you at? Hello?
I'm at the third slide itself.
Oh my God.
Yeah, third slide.
No, she's at the third slide.
Third slide.
All right. Okay, so we said dehydration or I've said that hyperhydration can greatly influence measured hemoglobin levels. Albumin can indicate hydrated status. If the concentration is below normal, this is called anemia. Anemias are classified by the size of red cells, the cells that contain hemoglobin in the vertebrates. Now, there is some disturbance over here. All right. Shanti, we are recording this lecture. So this lecture is going to be on YouTube in a day or two. So you can follow it over there too. Let's go further.
Okay, thanks.
Okay. We've said that delta chain. Huh? Yeah, so you got alpha 2, delta 2. The delta chain synthesis begins late in the third trimester, and in adults at normal is 1.5 to 3.5%. Fetal hemoglobin alpha to gamma 2 is the adult hemoglobin F is restricted to a lower population of red cells called F cells. However, the level of hemoglobin F can be elevated in persons with sickle cell disease and beta thalassemia. Yes, they've already seen them. All right. This is also, we said, what this has been done. Let's go further. So we come to uses of hemoglobin. Sandhya, are you hearing me? Sandhya?
Yes, ma'am.
Any discussion you would like to add a few points? Or Shanti, would you like to add a few points here? We are on slide 14 right now, where I'm concerned.
Oh, no.
It's okay?
Yeah, great going.
All right. So we come to the uses of hemoglobin. And, it's strange that sometimes when I ask a new technologist coming into the lab, what are the uses of hemoglobin? Why does your body need hemoglobin? And they give me a very blank look as if they don't know. And I said, come on, I said, this is now taught in standard 3 or 4,
Yep.
the use of hemoglobin and oxygen. And, yes, it carries oxygen. Now, that's strange. This is the new medical technology. And they pay through their noses for programs like this. That's the reason why I would like to do a podcast on medtech education maybe in a month's time. I had asked Chaya, but Chaya's in America till August to do it with me. And it's high time we did something about the medtech education that we have today. It's pathetic. All right, so we come to use of hemoglobin. Anemia is called microcytic if the red cells are small, macrocytic if they're large, and normocytic if they're more or less normal. Hematocrit, the portion of blood volume occupied by the red cells, is typically about 3 times the hemoglobin concentration measured in grams per deciliter. For example, if the hemoglobin is measured at 17 grams per deciliter, that compares with a hematocrit of 51%. And you divide your 17 by 3, you will get your red cell. And it's so strange. I have a student many years back called Salma. She's in the UK and she's working shifts, etc., etc. And she said, "Ma'am, thank you for telling me the relationship I had forgotten between red cell, hematocrit, and how the comparison is done. So this is the way it's done, right? You agree with me, Sandy? 17 grams compares with about hematocrit of 51%. And 17 divided by 3, what is it, 5 point something? That will be the red cell count. So this is very important. You should keep it in mind. Usually when we see the histograms, the first thing we look at these 3 parameters and see if they are normal. If they're not normal, then chances are there is a microcytic hypochromic type of anemia or macrocytic type of anemia and so on. You can take a look at the pictures on the right hand side. You've got those very small red cells, microcytic hypochromic. Then you've got the normocytic normochromic, more or less normal. And usually we compare the size of the red cell to the lymphocyte, the small lymphocyte. That's about 6 to 8 microns, you know. And then you've got the macrocytic normoblastic anemia on the right-hand side, where the red cells are large, and in certain anemias, then you will get this picture. So we come to estimation of hemoglobin. This is the only parameter assay in automated machines by biochemical principle. Can be done simply in the lab by Drapkin's method, using a calorimeter at 540 nanometers, green filter. The principle and methodology will be dealt with separately. Drapkin's reagent is used in measuring hemoglobin from blood samples. It comprises of potassium ferricyanide, potassium cyanide, and potassium dihydrogen phosphate as components. Potassium ferricyanide oxidizes hemoglobin to methemoglobin and then to cyan methemoglobin. It's measured at 540. Can be done on completely automated 3-part, 5-part, and 6-part analyzers. But I have a feeling on the analyzers, Sandhya, correct me, there is another principle, am I right?
Yes, ma'am. But it is optical.
Yes, yeah. So everybody doesn't have the ferrocyanide on the machines. So we come to some CBC results. Sandhya is going to talk to you more about this in her presentation. So you've got the platelet, you've got the red cell, and you've got histogram. So you can see how it goes. And reading the histogram involves a little bit of practice and mathematics. And each company has their histograms and scattergrams. And I think Sandhya is going to tell you a little more about histograms a little later. I asked her, what about WBC and platelets? So she says, ma'am, that will involve scatter plots, which we'll have to do separately. And I agreed with her. I think I have some students. Parul,
Yeah.
Did we go into this with you guys as
Yes.
well as Urvi? I think you should know. Urvi, familiar?
Ma'am, I'm sorry, but a little lost.
Okay, that's okay.
No, we went into this, though we didn't do the HbA1c at all at that time.
But now HbA1c has become routine. We'll talk of that. Of that. And you have the expert. You've got Dr. Shanti Naidu sitting with you. She'll tell you more about it. All right. So we've got other uses of HbA1c. It's a good measure of diabetic control over 90 days. So long-term control of blood sugar concentration can be measured by the concentration of HbA1c. Measuring it directly would require many samples because blood sugar levels vary widely through the day. HbA1c is a product of the irreversible reaction of hemoglobin A with glucose. A higher glucose concentration results in more HbA1c. And somebody like me, I have to be very careful about it. I hate doing it when they are doing it on me. And I haven't done it for quite some time. So uses of hemoglobin A1c, the test, because of its reaction is slow, the HbA1c proportion represents glucose level in blood averaged over half-life of red cells, typically 50 to 55 days. So it gives you the control of the last 3 months. And HbA1c proportion of 6% or less shows good long-term glucose control, while values above 7 are elevated. The test is especially useful for diabetics. Uh, Shanti? Shanti? She around? Because the American Diabetic Association has changed these normals a little bit, especially for a diabetic. And my nephew was down from the US about last month, and I had his parents' HbA1c, and when he was looking at the normals, he's telling me, he says, Ruz, is this what you'll follow? I said, yes, this is what we have as a normal level. But then I showed him some literature on the American Diabetic Association, where they have said that if the person is diabetic, up to 7 is considered normal, 7%. So I was quite happy
But
because,
that's the age group in that, Rose. There is an age group.
yeah.
7 is considered normal for a diabetic around after above 60 years or so.
Okay. So
That's
that's the
the
reason why, Shanti, I'm very happy. I keep around 6, 6.2, 6.3. So,
Yes, but 7 is acceptable in that age group.
right. And you know my age group, so I very much acceptable. Let's see what my new HbA1c, I'll be doing it in July. And Munira's after me for the quite some time to have it done, and I'm putting it off. All right. So we'll talk about that. So we come to heme synthesis. Now this is biochemistry. And I hope that at least my students are listening to me here. In their mind, they will revise what I've taught them because I think this was taught to everybody. So hemes are cyclic tetrapyroles that contain iron and are commonly found as the prosthetic group of hemoglobin, myoglobin, and cytochrome. Is the noise?
Someone's mic is on.
Someone's mic is on. Do you mind muting it, please? It's a little bit of disturbance. Thank you. So heme synthesis takes place in the cytosol and mitochondria of cells of the liver and bone marrow. The substrates are succinyl coenzyme A, glycine, iron in the ferrous state, and the product is heme. Hemes are a diverse group of tetrapyrrole pigments, being present as the prosthetic group of both the globins, hemoglobin and myoglobin. And you can take a look at the picture with iron in the middle. It's a cyclic structure. You've got the pyrrole ring, you've got the methyl part, you've got the propionate, and you've got the vinyl part. Okay. You try to follow on the right hand side instead of going through the circle. So this is the way it happens, right? Succinyl coenzyme A plus lysine. The presence of ALS synthesis will give you alpha amino ketoadipate, which in the presence of ALS synthesis will give you delta amino levulinic acid. Further, to give you porphyrin, biringen, protoporphyrin